Sometimes it's just a single step because grams is a base unit. Hello. Hello. Yes. Okay. Bye. So in doing this one, make sure convert the mass to grams. So this one, grams and milligrams. Anytime you have milli, milli gets the 1. Milli gets the 1,000, grams gets the 1. 0.62 divided by 1,000. Gives me the mass amount. And then I'm going to divide it by what? Nope, because look at what I want my units in. So you want to put it in grams per milliliter. So put your units in grams per milliliters. So I don't have to do anything with the volume. So it can just be as is. But if I want to know parts per million, what am I going to multiply that answer by? 1,000,000. So what'd you get? 2.48. So make sure you put that in the calculator, see if you get that. So there's the parts per million. So is this safe? Would this be a safe sample to drink? Yes. Why? Because it's less than 4, right? So see that the maximum fluoride in drinking water can be safely added up to 4 parts per million. If they add more than 4 parts per million, you can actually end up getting like white spots on your teeth because you can get like fluoride deposits. So that's why they don't do that. Nobody wants some white speckly looking teeth. Okay, so 4 parts per million, sort of the limit for it. So if you end up calculating, you always put your mass in grams, put your volume in milliliters, just divide those and multiply that answer times 1,000,000. That comes out as 2.48. So that is, yes, it would be safe to drink. So 2.48 is less than 4 parts per million, which is our limit. Okay, so you'll have like a threshold or a limit for whatever it is. Yeah, just change all of the sodas out. Oh, that's terrible. There, should be open now. Okay, so now the next one. So the other one to practice is doing a last dilution. So what was the original concentration of a glucose solution if you use 275 mils of this to make a 1.75 liter solution of a 5.25%? Okay, so in this one, this is a dilution question. So C1, identify what each of those units in the problem mean. Like, what do they represent? Concentration versus volume, initial versus final. Rearrange the formula so that you have the one unknown all by itself, plug in the numbers, and then assign those significant figures. So I give you 3 minutes to do this one. Okay, so in this one, what are you trying to find? So what is the original concentration? So that's C1. That's the one I don't know. So what is 275 mils? That's V1, so I know that. What is 1.75 liters? V2. And then 5.25%? C2. Okay, so C1 is the one I want to find. So what unit do I move? You're going to move V1, right? So dividing both sides by V1, that's going to move it to the opposite side. So now, in setting this up, if I plug in C2, 5.25%, times V2, which is 1.75 liters, divided by V1, which is 275 milliliters, do you see an issue? In order for units to cancel, they have to be the same. Okay, so if you're given like two, like I promise you the concentrations will be the same. They'll both be percent or they'll both be molar. Okay, if they're both percent or both molar, as long as you set them up, both of them cancel. You don't have to change anything with concentration. But with volume, see how one is milliliters, the other one's liters? So you can pick. It doesn't matter which one you change, but you got to change one of them. They have to both be in liters or both be in milliliters. It's not going to change your answer depending on which one that you convert. But just because this one's all by itself, I'm going to go ahead and do that one. So 275 milliliters, I got to put that in liters in order to be able to use it. Because liters and liters need to be able to cancel. So 275 ml times in a line. Remember that unit goes on the bottom. The unit I want goes on top. It's a single unit conversion. Milli always gets the 1,000. Liters gets the 1. So that's going to change this to 0.275 liters. Now I can put that into my problem. So you see now liters and liters can cancel. So just make sure that you check if you're doing a dilution, check the volume might not match. You've got to make sure that those units match or they don't cancel. So then putting this in, 5.25 times 1.75 divided by 0.275. I got, tell me if that's right, 33.4090991, and that would be percent. Yes? Can I keep all that? No. So go back to the original problem. 2.75 has 3. 1.75, 5.25, so pretty much 3 is our significant figures. So remember, you just start from the left, underlining the first 3 significant figures, the 33.4. So all the rest of it does what? Drops off. So that is the original concentration of my solution. And it's glucose. Okay, so just this one's like, it reminds me very much like a gas law. You just figure out what's the one I don't know, what does each of those units represent, rearrange the formula so that you get the unknown all by itself in the formula, then just plug them in. Double check to make sure that your volumes are the same units. If not, you're going to have to convert one or the other. And then just round with significant figures at the end. So just thought I'd do a couple more of those. The very last part of Chapter 8 is all about passive, well, really, it's all about what's called transport. So transport, and this is going to go into a lot of biology. So if you've had any biology, this should be something that's typically covered in like the second chapter and then covered again later chapters, especially if you do like the A&P. You've talked about this in your analysis because of kidney function. So transport is just the way that molecules are moved in or out of the cell. They are moved across barriers, because remember we said that the cell has that phospholipid barrier. So remember the phospholipids is where those polar heads are on the surface of the cell, the nonpolar tails tucked down underneath. There's a the polar heads are at the bottom or the inside surface of the cell. So remember that this creates this barrier. So things that are polar, things that are hydrophilic, things that are ionic cannot just freely pass through. So in order for the cell to get things in and out, most of the time, these molecules, the ones that are polar, the ones that are ionic, they have to have some kind of channel. And so the channel is a protein. And those channels like go all the way through. So if you remember that image of the cell membrane where we talked about how the phospholipids make the barrier, and then you've got those integral versus peripheral proteins. So that channel is going to control what goes in and what goes out. So in osmosis, osmosis is really talking about the drive for water to try and balance them. In fact, you have channels in the cell membrane that are specific to water because water is polar, right? So water is hydrophilic. Water can't pass that barrier, but water can move in and out of the cell. And the goal is really to try and get the total solute concentrations to be the same. Since your body is roughly 2/3 water, so we're more than 50% water, water is that universal solvent. So water dissolves. It's the solvent that dissolves all the salts. It's the solvent that dissolves all of the nutrients, the waste, gas molecules like carbon dioxide are dissolved in it. So there's all of these solute particles. Many of them can't pass the membrane barrier. So water can. So if you've got a lot of sodium, Na, lots of sodium outside, and just a few inside, do you see that there's a concentration difference? So there's a lot of sodium outside of the cell, less sodium inside of the cell, but the sodium, because it is charged, can't just pass through. So it would like to go in, right? So sodium would want, by just the process of movement, the sodium would like to move into the cell, but it can't because of the barrier. So the water tries to do this to balance it. So which way would the water move? It's going to go out of the cell. So it always moves from areas, if there's less solute and more solute, if there's a concentration difference, water always moves to the more concentrated regions. Because in theory, inside of the cell now, there is a lot of water molecules, H2O, a lot of water molecules compared to the sodium. Outside of the cell, comparatively, there's less water molecules. So the more concentrated something is, the less water it has, right? So a 1% salt solution has 99% water. A 20% salt solution only has 80% water. So the more solute, the less water. So water tries to move towards areas where there's higher concentrations, and they can actually use a channel to do that. So water has this ability to move in and out, and that movement is called osmosis. Now, optimally, your cells should have equal concentrations. So optimally, the amount of sodium inside and outside of the cell should be the same. So if we had a solution, typically it is 0.9% sodium chloride. Inside of the cell, 0.9% sodium chloride. So if you were normally hydrated, not dehydrated, not overhydrated, so if you have the correct amount of water in and out of the cell, then they say that's balanced. So the term they use for that is that it's isotonic. So an isotonic, iso means equal. Tonic really refers to solute, even though it's kind of weird because it's talking about movement of water, but it's really because of the presence of the salts or solutes. So if we have equal solutes inside and outside of the cell, then water, if water moves through the water channel, water moves in and out at the same rate. So you can have water moving in, but water's going to also move out. So that means there's no net change. It's balanced. Isotonic solutions, if you're giving a patient any type of IV, it's very uncommon to use anything other than an isotonic solution. So you give them 0.9% sodium chloride. So that's in a normal lactated ringer's bag if you're just running saline. So if they're running any kind of IV, they want to give you something that would be balanced with any of the cells in the body, any of the blood cells floating in the blood, and then also in other tissues. So if you have a person that drinks large quantities of water, that, in theory, could actually dilute the blood and cause the salt concentration outside of the blood to go down. Okay? So, like, we're not talking like you drink a liter of water. So this would be somebody that drank like 4 of these in like 20 minutes. Like, would really make you feel nauseous. Okay? That's one of the side effects of like that hyponatremia, low salt, because it gets too dilute. But if a person drinks a large quantity of water, that water gets absorbed through the lining in the mouth like glucose can. You can absorb it through the mucosa. Water gets absorbed through the stomach. So it doesn't have to get all the way into the small intestine. It gets absorbed really quickly. So if you have this rapid change of water influx, now think about this cell. So I said, so the cell normally would have, we'll just put like 3 in, 3 out. Right? And the amount of water then is also balanced with it. So it's in that isotonic condition initially of that 0.9%. But now if a lot of water comes in, now compared to, that's H2O, compared to the water, the salt molecules, do you see that now it looks like there's more water outside? So that is going to dilute the salt. So there's twice as much water, so that salt then is lower in concentration compared to inside. So they would say that this is now hypotonic. The term hypo means low. Tonic is, again, those solutes. So this is if you end up with a low solute concentration. Low solute because of that influx of water. But water then has this difference. The salt that's inside now is in higher concentration than the salt outside, and the salt would like to move, but it can't just freely pass through the barrier. But water always has those channels, so it has the ability. I think the noise can open it again. I feel like there's better airflow in here. Nobody has complained yet about my yelling, but okay. So in this, because it's not isotonic, because it's not balanced, water is going to move to try and balance it. And it does this automatically. They call this a passive process. It is a type of passive transport. Passive transport always has 2 criteria. 1, passive transport never requires energy. So there is no energy required. The cell does not have to do anything to get this to occur. It just will happen. Osmosis is always movement of water. Or the solvent. Water is going to move from where it's at high concentration to where it's at low concentration. So if you look at the image, where is there more water molecules? Inside or outside? Outside. So this is going to push water in to the cell. Through those water channels, and what ends up happening to the cell? It swells. So the cell will actually begin to swell. Worst case scenario, it could actually lyse because there is a limit to how big it can get. So the one to remember, if you remember in lab, when we did the osmosis and diffusion lab, you took the piece of potato and you put it in distilled water, which had no salt in it. So that's like the highest possible amount of water outside of the potato. So really the potato represents the cell. So water moved in, and how did it feel? How'd that potato feel? The one that sat in water.
It got hard.
It got hard, okay? So when you picked it up, it was really firm. You could hardly bend it. When you weighed it, what happened to the mass? The mass went up, right? So it actually pulled in water, increasing the mass of that potato. And so it actually made it firm, hard. It actually gained mass because that was all from the water. That is a hypotonic condition. And I will tell you that this is a risk if you have a patient that's dehydrated. It's rarely a problem. People just don't go around drinking gallons of water in a half an hour, okay? It's just, there's something a little strange if you're like wanting to do something like that. It's not going to make a big difference in your life. Like, it's much better to spread your gallon of water out over 24 hours instead of trying to drink it all at one time, okay? Because it really puts a lot of stress on the poor little kidneys trying to filter the excess out. This, though, can legit be an issue for somebody that is dehydrated. Because if you're dehydrated, then what's happened? Dehydrated, their water levels are already what? Low. So your water levels are already low. So your levels are low. So bringing it back too rapidly into balance can actually trigger this hypotonic response. So think like when you look at this molecule, when you look at the little blood cell, just pull out like on the outside of that cell, take away like 4 of those water molecules. So see, then that water is already low. So when you then replace that water too quickly, that could cause water then to shift into the cell and cause some swelling. Worst case scenario, this can actually cause cerebral edema, because this can cause water to shift out of the blood and into cerebral spinal fluid, which then puts pressure on the brain. The person then, it suppresses nervous system function, so they can actually go into it like lose consciousness.
Does it matter if the water is hot or cold? Because I've always heard like if you go outside and you're dehydrated, never eat it, like don't drink cold water.
I guess it would have to be very cold water. Because as soon as it hits your mouth and goes down the esophagus, it's actually absorbing warmth from all of those surfaces. So it goes pretty quick. I mean, some people talk about like getting brain freeze from like having a Slurpee or having like, like a slushy kind of thing. And that's actually when it hits the roof of your mouth, can actually trigger that temperature transfer. Have you ever gotten one of those? Like it's just way too cold, and then you're like, oh, okay. That is actually because of temperature transfer from your mouth up through the bones and up, and then it like causes a temperature shift in the brain. It could create that sensation of pain, but it's really not. It would have to be pretty cold, like icy, kind of like a slushy type of thing, for it to be more of a concern. But what did you, when you were sick, if you were sick, vomiting, everything else, when you start back feeling a little better and you want something to drink, what do they always tell you? Sip it, right? Don't drink it really fast. Don't you're like, I'm thirsty. Just take sips. That's really the reason. It's not an old wives' tale. If you're dehydrated, you want to rehydrate slowly, not rapidly, because influx of large amounts of water can cause fluid shifts from the fluid of the blood into the blood cells, from the fluid of the blood into tissues. So it could cause swelling or edema to occur, all because of that shift and change.
Is that why you're not supposed to, like, they've always said when you're playing sports not to be, like, jumping down?
Yeah, well, and that sometimes can cause, like, cramps and that, right? So if you have a lot of fluid, like, going into the small intestine and you're exercising, your body's not wanting to do a lot of digestion. So it kind of shuts down digestion because of the exercise. And then that can lead to the cramping that people, like, complain about. This is hypotonic. So I would say I would be more concerned about this from a dehydrated patient. So if you're taking care of the little 4-legged animals, have you ever had a patient that, a little puppy or a dog where they lift the skin and it stays tented? Yeah. So that's like, you can see that if you see this in a person, oftentimes you will see that the eyes look kind of sunken. The skin will look almost like flat, like it's sort of saggy looking, and it's really because of loss of fluid in subcutaneous tissues, like that kind of collapses. If you're talking about a baby, their little fontanelle will be sunken in if they're dehydrated. So dehydrated patients, it's much more of a concern that you want to hydrate slowly. This is why they hydrate with an isotonic salt solution rather than trying to hydrate with just pure water, to try and like keep that balance, bringing fluid levels up kind of gradual over time.
Is that why they make it like, they make the, when you have an IV, they make it really slow?
So it's given slow. One, you don't want to blow out a vein, right? I mean, if you were like if you've ever seen like in the TV shows where they're like, squeeze the bag. I'm like, oh, that's really bad for your veins. Because if it goes through in a lot of force, your veins are really like flexible. They're not used to that. They're not like an artery, which has a lot thicker, more reinforced wall. So veins are much more apt to end up being damaged from stuff like that. So yeah, that's when you're like, yeah, there's some things that they do. It's for the drama, you know, like the drama effect of the show. Nope, nobody likes that. Nope, no necessary. Yeah, physiological saline would be giving saline solution. And they can give like, so they would give like D5W is one that's common. That's a physiological saline. That's just dextrose 5% in water, D5W. And all that means is it's actually giving you a fluid that's going to help get your blood sugar right about 80.
So that's what
right?
lactator ringers are?
Lactator ringers typically have glucose in it and also has some other salts, but it's physiological, right? So it's going to be isotonic. And the goal is then we're just going to bring your fluid back to that isotonic level, right? So the other one that happens is hypertonic. So under hypertonic conditions, this really does happen if somebody gets dehydrated. So when you get dehydrated, so you're a little kid, you're an adult, you've got the flu, so that means that you have a fever. When you have a fever, you sweat, but you don't notice it because it evaporates, but you're actually sweating. So sweating is a loss of fluid. If you're vomiting, you're losing fluid. If you have diarrhea, you're losing fluid. So all of those are now depleting, and if you're not able to keep fluids in, then that means that your fluid output's going to vastly overcome the fluid input, and you can end up dehydrated. So thinking about a cell that has that condition, so your poor little red blood cell, so we'll just put a couple of NaCl's inside and outside. I'll add the Cl, NaCl. So this should be normal, right? Should be equal amounts inside and outside, concentration-wise. But now I have inside of the cell, the water balances the salt. So like for one salt, they're equal. But over on now, because I have lost water, so now when I've lost water, so if you look at the water concentration here, do you see that now, because you've gotten dehydrated, the amount outside, the water concentration's gone down? So now where is water at higher concentration? Inside the cell. So there are more H2O molecules inside, and this is the premise then. Water can move. It's just moving to try and rebalance that salt, to try and get the concentrations. It can't get the concentrations to isotonic, but if it can rebalance it, then that's a little more evened. So some of the water then begins to move out of the cell to try and balance out the loss of water outside. So then what happens to the cell? It starts to lose water, right? So then the cell starts to get all shrunken and shrivelly looking. Like a raisin. Yep. If you've ever done this in the microscope, it's pretty cool to watch them, but you can actually see them shrink up so that they get real spiky, red blood cells do. That loss is all because of this hypertonic condition. So remember, this means that there is a high solute, not enough water outside of the cell. And this condition is common with somebody that's dehydrated. Have you ever been dehydrated? Anybody ever been dehydrated? How did you feel?
Horrible. Every time I drink something,
Do you get like really dizzy? Right? You get almost like out of it. You're like, I don't really know where I am now. That is because literally you begin, if you start to lose fluid, then that means that the blood volume begins going down. Now your blood pressure starts going down. You also can then pull fluid out of cerebral spinal fluid to try and maintain blood pressure. So now your poor little brain doesn't have as much fluid. And so that's where all those neurological changes begin to happen. So it can really kind of like start this downhill spiral. But people like describe being dehydrated as just being like so weak. I'm like, I really can't move. I'm just going to lay here now. But that's all because of that fluid loss.
I got dehydrated one time.
Mm-hmm. Just like that's like, but at least the orange juice like has a lot of sugar in it. It has got the vitamin C in it. And so it's not like drinking pure water, right? Because you're drinking something that has solutes in it. So in that, as it gets absorbed, it's not like you're drinking 100% water. You're drinking something with some solutes in it. That's why they like Gatorade, to try and hydrate, because it's a little more isotonic and it's not going to cause that fluid shift so quickly.
every time I try to drink something, I just throw it out.
Yeah, that's the worst case. And that's when a lot of times they'll hospitalize people because they get so dehydrated because they can't keep anything down. And so then, but giving you an IV, we can replace that fluid and you're not going to lose it. And then they can give you the antipyretics and stuff to like calm the nausea. So there's a lot of pretty decent like cancer treatments or cancer meds that they give people that like can't stop vomiting, because that's what happens when you go through cancer treatment. So there's a lot of emetics, they're called, which is just like stop you from being so nauseous. That really does help to try and balance it out. So here's a pretty cool picture. I like this one. If you've done this in lab, then you've seen these changes that occur. So the bottom one on the far left shows isotonic conditions. So this shows what a red blood cell looks like if it's in a balanced salt concentration. So notice it looks almost like an inner tube. It's kind of like a donut without the hole in the middle. So they say it's a biconcave disc. So that is isotonic because it's equal salt. So if any water goes in, water is going out at the same rate, so there's no change in the shape. But then if you put it in that hypotonic condition, so remember that this would be very low salt in the solution, very high water content. Low salt has more water, and so that would cause water then to move into the cell, and see how the cell gets really baked like a ball, so it's completely swollen. Hypertonic, though, is if you end up having the solution has a high salt. If you're dehydrated, you don't have enough water. So that means the salt becomes more concentrated compared to what the water concentration should be. And this actually causes water to come out of the cell, and the cell ends up looking like all spiky and raisiny, bumpy. The whole reason, if you remember like cytoskeleton, the whole reason it ends up being spiky is your red blood cells have like a little protein framework that normally maintains that biconcave disc. But if you pull all the water out, it kind of like shrinks around that framework, and that's what all those spikes are. So the spikes are kind of like the little cell's skeletal structure to maintain it. It's pretty interesting to watch those changes. You can just do that like by changing the salt concentration. That's osmosis. Okay, here's another way to look at osmosis. So do you see in that cell membrane, outside versus inside? So notice outside, the green and yellow balls, these are solutes. So both green and yellow are solutes. So when you look, do you see there's more to the right? More green and yellow balls, less green and yellow balls inside. So you would say the outside there has a higher concentration. The inside has a lower concentration. So think of the kind of like this area. This is really the water, right? And this is the water. So the blue or blue-gray areas, that's the water. So can you see that there'd be like less water on the right side compared to the left? There's more open space on the left side of the membrane. So that would be the higher water concentration compared to the amount of little blue area on the right. And that's the reason that water goes through. So water will pass. They don't show it, but remember that there is a water channel that is going to span the entire membrane, and it's open and it allows water to move in and out. Water can't move all by itself through because it's a polar molecule, and the phospholipid barrier is a block, but there actually is a water channel in all of your cells to allow for this balancing. One person, I've heard a lot of like nursing students especially, they said that they've always taught like water always follows salt. So do you see where you have a high concentration to the right? So water always goes to areas with higher salt. And the goal is it's just trying to dilute it. It's just trying to make the total concentrations in and out the same. So that term that water always follows the salt. If you have a high salt condition in one area, water's always going to try to move in to dilute it, which means it goes from high concentration to low concentration of water. Okay, so just another way of kind of looking at it. This is why they try to give what they call physiological saline. So like I said, 5% glucose, 0.9% sodium chloride. They like to give physiological solutions. Physiological just means living systems. Okay, so this would be the normal solution in a perfectly healthy person. So that is the common type of solution that they're going to give, rather than giving a hypertonic or a hypotonic condition, because it can cause fluid shifts to happen too fast. I only know of one time that they'll actually use a hypertonic condition. They will use a higher salt, higher than 0.9%, if a patient has cerebral edema. So if a patient has swelling in their brain, maybe they've had a TBI, swelling in the brain, this is almost like a last ditch effort, but giving them high salt can actually help pull fluid out of cerebral spinal fluid and help to decrease that pressure. But it's very rare. Typically they stick with anything that's physiological. So you start with something that's balanced rather than something that's above or below what it should be. So then the other kind of passive transport. So water moves by osmosis. All solutes move by diffusion if it's a passive transport. So this is also a passive transport. And this first one, that's called simple diffusion. Simple diffusion is when molecules move, or solute molecules move from high concentration to low concentration until the concentrations are equal, right? Osmosis is the same. If something is high and low, movement happens until they become the same, and then transport stops. So their example they give here is like if you have a glass of water and you add a drop of green food coloring, notice over time, the entire container ends up turning green. So that little drop was in high concentration, but as soon as it hits the water, it's going to want to spread out. So remember I told you that the disorder is like the natural tendency of things. Things like to spread out. Things don't like being in high concentration. They'd much rather be uniform concentration throughout. And so this helps to do that. So molecules are going to move. Not water, not solvent. This is solute. From high concentration to low. Until concentrations are equal. So just putting sugar in your coffee, in theory, if you put sugar, most people can't wait this long, but if you put sugar in your coffee and you just let it sit there, over time the sugar would totally dissolve, mix throughout the coffee, and it would taste the same at the top or the bottom. But you don't have time for that, so you speed it up by doing what? Stirring it. So stirring just speeds up the diffusion to evenly distribute the sugar throughout the coffee. That's all just movement of those solute particles. So there is this simple diffusion. Simple diffusion in terms of cells occurs when molecules move straight through the cell membrane. The only way they can move straight through the cell membrane is if they are nonpolar, because remember that phospholipid barrier doesn't let polar things move through, doesn't let ionic things. Hydrophilic stuff can't pass. Hydrophobic can. So nonpolar molecules, oxygen, nitrogen, carbon dioxide, the gases, because they are one, very small, and two, non-polar, they can actually diffuse through the cell membrane directly into the cell. But they only move from high concentration to low concentration. They'd never move the opposite direction. So they call this simple diffusion, not passive. It's really simple diffusion because it doesn't need any help. So when you take a breath, air flows in, oxygen in the air sacs of the lungs is in higher concentration than the oxygen that's in the blood vessels surrounding the air sacs. So we have a high concentration, low concentration. We have a lot of membranes between. You have the cells that line the air sacs of the lungs. You have the cells that line the capillaries of the blood vessels, but oxygen can just diffuse straight through. But it's only going to move from high concentration to low concentration. So pulls in, moves through in the lungs. Oxygen in the lungs is going to be in high concentration. So now you've got the cells that line the air sacs, and then you've also got the cells that line the blood vessels. So it's actually like 2 cell layers. So you would think like that seems like that would be pretty hard, but it's because it's non-polar, so it just is able to pass directly through. Oxygen is going to diffuse into the blood because in the blood, it's at low concentration. So that is how oxygenation occurs. Doesn't require any work by those cells.
Would anemia be an example of any of these?
So anemia is if you don't have enough red blood cells. So remember that in the blood, oxygen's carried by red blood cells. So if you don't have enough red blood cells, then that's going to limit how much oxygen can diffuse. So that's why your blood ends up having less oxygen and you stand up really fast and you get dizzy and yeah. Or you're like, come on, let's go. And you're like, I just can't. Okay. It's because you don't have enough oxygen to power muscles. So that can affect this, but this still is a natural occurrence. Passive diffusion, it's simple, doesn't require any kind of work. Now, what's interesting is carbon dioxide goes the exact opposite direction. So carbon dioxide is dissolved in plasma, but it's in higher concentration because your cells make it. So your cells make carbon dioxide and it diffuses out of the cells and into your blood. And when it gets back to the lungs, carbon dioxide in the blood is in high concentration because it is the waste molecule that you make when you break down glucose. So your cells break down glucose and make lots of carbon dioxide, which it then allows to diffuse into the blood where it's in tissues, it's at low concentration. Now the concentration goes up, blood moves along through, goes back to the lungs. Carbon dioxide's in high concentration in the blood, but in the air, carbon dioxide's in really low concentration.
Yeah, I was gonna say, don't we like breathe out?
Mm-hmm. Yep. So you do exhale this because if it's in low concentration in the air, this is going to actually go the opposite direction, high concentration to low concentration.
Okay.
So carbon dioxide diffuses out of the blood and into the air in the lungs and you exhale it. So when you bring in fresh oxygen, diffusion happens, oxygen moves in, but carbon dioxide's moving out. When you exhale, you get rid of it.
Okay.
As long as your lungs are normal, as long as your lungs aren't damaged, this is never an issue. Patients that have emphysema, I don't know if you've ever taken care of anybody with emphysema, with bronchitis, with pneumonia, chronic obstructive pulmonary disease. A lot of patients that had COVID, scarring in the lung tissue can severely restrict this. Because remember this layer? So what happens when this layer ends up having, you can like put almost like this physical scar tissue barrier between. That can limit diffusion. So healthy cells, just 2 little flat, thin, remember those simple squamous cells, 2 flat layers, oxygen can move pretty easily through. But when those cells start to get big and inflamed, when you start having scar tissue, so now you've literally got like a tissue layer, that severely limits how well oxygen and carbon dioxide exchange can happen. And that can then lead to like permanent effects where they can't get the oxygen as well and they can't get rid of carbon dioxide and it ends up building up in the blood. So some of those respiratory conditions really affect this type of diffusion in the lungs, you know. No, it's like that's like one of those chronic issues, respiratory issues that person, other than doing, I have heard of some COVID patients that have had lung transplants where they've actually gone in and transplanted lungs because their lungs are so scarred that they're like, that means like you're talking about like how you feel dizzy with the anemia. If you can't get oxygen, everything is in slow motion because all of your muscle cells are like low on oxygen. So they cannot make a lot of ATP. So like eating is exhausting. You know, like just physically chewing, any kind of movement. So basically you like have this really, your world and your space gets really small. Respiratory issues can affect this movement. So there's one other. Facilitated is talking about molecules that are actually polar or ionic. Can these pass through the cell membrane? Can polar molecules pass through the cell membrane by themselves like oxygen can? No, because remember that the cell membrane has that phospholipid bilayer, and that's like a wall to anything that's hydrophilic. Anything polar and ionic cannot pass straight through. You have to have a channel. So there are solutes, and one of them we've actually talked about. Solutes like glucose, solutes like all of your salts. These cannot freely move in and out. They have to have a channel. Waste molecules. And that's why they call them facilitated transport. They have to have a channel. So that means that there has to be an integral protein in the cell membrane wall that allows certain molecules to pass through. Those molecules only move from high concentration to low. So when this door opens, I told you disorder is the natural thing. So when the gate or the channel opens, if there's a lot of these molecules here, they move in. If there's a lot of molecules here, they're going to move out. Okay, so they'll move in or out to try and balance the concentration because it's always high to low until we reach this equal concentration. So glucose, remember we talked about glucose. So kind of like this picture, the first one shows oxygen and how oxygen moves. So this is not in the lungs. This is talking about in the tissues. So like surrounding where your muscles are, where your skin cells are, thinking about your brain cells. So the actual living cells, not just in the lungs, but oxygen then comes out of the blood and diffuses into cells because if it's at high concentration, that oxygenated blood gets passed to cells at the tissue level. So that then your cells can use it to break down glucose and make plenty of carbon dioxide and energy. So that's this example. Notice high concentration to low. And also notice that it looks like it goes straight through. No helper required. That's your simple diffusion. Facilitated though, see the blue channel. So facilitated diffusion, good example in this one, glucose. So when you eat, your blood sugar goes up. So now there's more sugar outside of the cell than in. What is the key to open this channel? What do you have to have for this channel to open? If you don't have it, then this channel stays closed and you can have really high blood sugar numbers. Insulin. So remember insulin here. Insulin is the key. You have to have insulin for this channel to open. Otherwise, the door stays shut. So I think of it as being like a key to open a door. Without it, it stays like this. And that's why you can end up with glucose, tons of glucose in the blood, but the cells can't get it because insulin is the signal that there's plenty of sugar in the blood. Go ahead and open the channels. Insulin's present. Glucose now goes from high concentration to low concentration until concentrations are equal. So all the glucose won't go in. Enough glucose will go in so that now the blood sugar concentration and the sugar concentration in the cell are equal. And then it stops. So even if insulin's present, it can't make more go in. It's just going to move from high to low until concentrations are balanced. All right, now look at this last one. So this last one, this is not passive transport. What do you see happening? Okay, so it's going out. But in fact, diffusion can move things out, like waste molecules move through channels and move out of the cell. They just still go from high concentration to low. So we could have things moving out or in from high concentration to low, but this one is what? Right? You see that one, energy is required. So that is different from passive. Why is energy required? What is happening in that last one? We are moving molecules from what? Look at the arrows compared to the other two.
There's 2 arrows.
We're moving from, it's in to out, but we're moving low to high. Do you see that? There's low concentration. There's only one little blue ball inside. There's 6 outside, but we're still moving that one. Do you see that that's opposite? That that's moving things from low concentration to high concentration. This is like cleaning the house, right? This is putting things in order, putting things all on one side. This is opposite of the natural order of things. Most things like to spread out. This is actually taking and putting molecules specifically on one side or the other side of the membrane. This is why this is called active transport. This never happens on its own. This always requires energy. So why do it? Right? Why waste energy to do this? Does anybody remember in their A&P class when they had things that ended up getting pumped into an area so they were at high concentration? So the term gradient was used or imbalance was used. So listen, so does anybody remember from A&P hearing about pumping molecules from one side to another, making an imbalance, creating a gradient, making a high concentration of ions on one side or the other side of a cell?
Is it the pump that has like the 2 molecules?
What like this? So in like sodium and potassium, this happens. What is that involved in? Oh, you guys! Come on, Andy. I know you've had it.
Hey now.
Come on. Sodium gets pumped out of the cell. Potassium gets pumped into the cell to create an imbalance. What cells do that? This is how you send a nerve impulse. So in order for nerves to send impulses, they have to create an imbalance. So this is why people say, like, I've been studying and I'm starving. Well, you've been burning a lot of ATP by generating a lot of nerve impulses. So if you use a lot of ATP, that means you've actually broken down a lot of glucose. So just sitting and studying, really like thinking hard, you actually do end up decreasing your blood sugar. And you're like, I'm really hungry. I've been studying all afternoon. That is a legit thing. Okay? Because it requires energy, because it doesn't happen on its own. So it's the only way you can send a nerve impulse is by your nerve, the axons, they pump sodium outside of the cell, potassium into the cell to create an imbalance. And then when that nerve gets triggered, those ions move in and out of the cell. Sodium moves in, potassium moves out, sodium moves in, potassium moves out, right? To be able to send that impulse along the length. And as soon as it goes past, it resets. So every time your nerve resets itself, it uses ATP. So it has to do this constantly. So it's always like ready. Your nerve cells are always like ready for an impulse. They always have to create that gradient. Another one. Have you ever heard about, putting you all on the spot, have you ever heard about calcium having to be stored? because when calcium is released into the interior of the cell, it triggers interaction of proteins. Anyone? That happens when what? What cells do that? Lord of mercy. Calcium. So this is when acetylcholine is released, stimulates a receptor. The receptor sends the stimulus down through the T tubules, which then causes the sarcoplasmic reticulum to release calcium, which then allows actin and myosin to interact. That is involved in what? What cells do that? Lord, I'm going to talk to Mr. Awadon now. Muscle contraction. So this is all muscle contraction. So in order for muscles to contract, your cell, those actin and myosin fibers have to have access to calcium. But you don't want your muscle cells to just randomly contract, or you'd be like spasming all the time, right? So when the muscle's at rest, all that calcium gets pulled out of the cell interior and stored in the sarcoplasmic reticulum, which is really like an endoplasmic reticulum. It's just in a muscle cell, so it's got a fancy name. But that's really all that it is. And so the stimulus is what triggers its release. So the nerve stimulates the muscle then dumps that calcium into the cells so that actin and myosin can interact, and that causes those the muscle cell to shorten. So that's involved in that contraction. If you can't do this, no contraction happens. If you can't put the calcium kind of away, put it into an organelle so that it's not around the actin and myosin, then you would end up with tetanus. Your muscle cells would stay contracted permanently, right? That's what happens in rigor mortis. So that's what happens after you die, because your muscle cells stop putting calcium away because they don't make energy anymore, and it's an active transport. You have to have ATP to do it. So there's active transport. It's not something that happens naturally, but your body uses it, your cells use it for very specific jobs, okay, to try and trigger certain activities that allow our trillions of cells to be able to move, to respond, to sense changes that go on. So that one's very different than the passive transport types, which are really just trying to keep things kind of balanced in and out. Balancing water, balancing salts, balancing nutrients. Okay, so the last topic in this chapter is an application of this. So this is about the kidneys. So one, you have two kidneys. Where are they? They're anchored in the back wall of your, what cavity is that called? Your abdominal pelvic cavity, Lord have mercy, located in the back wall of your abdominal pelvic cavity, right? So remember, like the diaphragm is the top, so all of this abdominal pelvic cavity, and they're really like anchored in the back wall. Typically, they've got a little bit of fat around them, so they're kind of cushioned in there. Microscopically, your kidney's big job, they're the filter organs. Their job is to filter out any solutes and water that you don't need. They have microscopic filters that are called nephrons. You have 1 million nephrons per kidney. So there's 1 million. So like if you look at them microscopically, so the nephron's got the little glomerulus and it's got these little tubules that are there. So the goal is the kidney filters the blood. It removes almost all of the fluid, not all because then the blood would get solid, but it removes almost all of the fluid from the blood. It puts back all the nutrients, and it puts back a balanced amount of salt and water into the blood, and the blood leaves and it's been filtered. But what it keeps, it does not retain wastes. It does not retain excess salt. It does not retain excess water. So these things, as this filtered fluid moves through, what you need can get picked back, pulled back into the blood. What you don't need stays in these nephrons and eventually collects and becomes urine. So you can filter almost an entire cup of this per minute. So it's like, because you got 2 million of them. So high function, lots of fluid gets filtered out. In fact, your entire blood supply, so your entire blood supply with functional kidneys, it is filtered about 35 times a day. Every time it passes through the kidneys, wastes, salts, water that you don't need, those things get pulled out and don't end up going back in the blood. That ends up becoming urine.
Is this different from administration?
No. So that's more thinking about like red blood cell volumes and hematocrit. So this is just so you've got the primary waste molecules. Waste molecules are urea, uric acid, and creatinine. These are mostly nitrogen waste. So when you eat protein, there's nitrogen in protein. If you use protein for energy, you can't have nitrogen. Your liver converts nitrogen waste into these waste molecules so your kidney can get rid of them. They're filtered relatively easily. Urea, uric acid, and creatinine. Those are the 3 big waste molecules. But you don't want these to build up in the blood. So the kidney's job is to flush these out, along with excess salt and excess water. In fact, you only need a total of 500,000 kidneys, 500,000 nephrons. So you only need about 25% of the nephrons that you're actually born with. So you need at least 500,000 nephrons. If you have less than 500,000 functioning nephrons, then you're described as being in kidney failure. Okay? So kidney stress, low kidney functioning, kidney failure, those are all going to be described. This is why, since you really only need 1 kidney. You start off in life with 2 million. You start off with 2 kidneys. That's why some people can donate a kidney. You can donate a kidney as long as both of your kidneys are happy and functioning and fine. You could donate a kidney and you'd still have twice as many nephrons as you need. As you get older, you do start losing nephrons. What was the disease that I said ends up causing kidney damage? Because of really thick blood. Hypertension can be an issue. Diabetes is really the big one. And that's because, remember that hyperglycemia. These are filters. So think about if this fluid is super, super thick, when it tries to go through these very, very small blood vessels, those blood vessels can just rupture. And if the nephron is damaged, it just shuts off. Okay? So these don't grow back. That's why you're born with 4 times more than you need. And most people never have an issue their entire life.
So that's why most diabetics are on blood thinners, right? Because of that.
Because of issues with blood clotting, too much pressure, all of that, hyperglycemia really does create hypertension. So it's not necessarily going to keep those nephrons, giving them a blood thinner. If the blood is too thick because of sugar, it doesn't flow well. It flows like syrup. And so that puts too much pressure on the walls of those vessels. And if they rupture, they just shut down. So if you have less than 500,000 nephrons, now you're not filtering as well as you should. And so you may end up having to have dialysis. Okay? So I'm going to see if it'll let me do this, because last night it would not. Hemodialysis. So can you see this? There. I think that looks pretty good. Stay. Okay. Can you tell that there are 2 tubes attached to this man's arm? Okay. Can you see that there's a tube going up and then there's a tube coming down? They actually have 2 needles in them. Okay, so there is a port, and this is drawing blood out, and then there is, well, it's usually, yes, it's this one is drawing blood out, and then you'll have another one that's actually putting blood back in. So in hemodialysis, because the kidneys are not filtering, what builds up? 3 things. If the kidneys are not functioning, what are the 3 things the kidney's job is to remove? Wastes, excess salt, and water. Right? So if your kidneys are not functioning, those 3 things start to get all out of whack, and that can end up affecting normal function. It can cause your blood pressure to go crazy. It can cause other organs to shut down because those waste molecules, they need to be filtered out because they're toxic in high concentration.
Can your, like, after you're on dialysis, does that, like, heal your kidneys? Like,
No.
It just,
It's an alternative to normal kidney function.
Okay, so if your kidneys are, like, failing and they're failing, you have to get out all the time?
Mm-hmm. So has anybody ever taken care of anybody with dialysis? How many times a week?
So I have some that go 3 times a week, some go 2 times a week, but I have some patients that I have to sit with because they're trying to pull the cords out, and they're just combative patients. So,
Mm-hmm. And how long does this take?
some of them, it depends. Sometimes they'll want them for like 3 hours, but it's usually like 5 to 6 hours.
3 times a week, somewhere between 3. I always hear about 4 is about an average number. Takes about 4 hours to do. So you go on Monday, Wednesday, Friday in the morning, and you get there at 8 and you don't leave until 12 or 1 o'clock. Okay, so 4 hours each time. So this tubing, the tubing coming out, so notice here, so here's the one where the blood's coming out. This blood coming out is going to be high in waste, and it may be high or low in water and salts. Usually it's in excess. It really depends on what you've been eating, right? So if you've been eating a lot of salt, then there's going to be a lot more sodium in your blood. If you're somebody that's like eats a really low salt diet, then it might not be as bad. So it really depends, because your kidney's job is to get rid of all the extra stuff that might be floating around in the blood that you ate that you didn't really need. And so then that's excess. So it really depends to a large extent about diet as to what these levels are, but it's definitely not in balance. So the patient goes to the dialysis center, they hook up 2 ports, they draw the blood out, and it goes into this dialysis machine. So this machine circulates the blood through this tube, but the tubing in the machine is a little different. The tubing in the machine is like that dialysis tubing that we used in lab. So the tubing in the machine has microscopic holes in it. So it's got small little holes that are big enough for water to move, holes that are big enough for salts to move, holes that are big enough for waste to move, but blood cells can't come out. So the red blood cells, white blood cells, and platelets, and even those proteins, remember I was telling you about there's proteins in the blood to make a colloid, those are too big. Those don't come out. So all of your red blood cells, white blood cells, platelets, proteins, plasma proteins, those all stay in this tube. Surrounding this tube is dialyzing solution, or dialysate is what they call it. Dialysate, this is isotonic. So iso means it's equal, right? So it's isotonic. So surrounding is this tubing is this equal salt, water, nutrients. And so what ends up happening? How does diffusion and osmosis work then? In the blood, what's going to be high concentration? Wastes. Wastes begin to diffuse out of the blood. There is, if you look at this dialysate, do you notice that there's no waste molecules? There's no waste molecules in the fluid surrounding these tubing. Inside the blood, in this tubing, is where the high waste molecules are. So this is going to go from high to low. So high waste molecules will begin to diffuse out of the blood. Keep diffusing, keep diffusing, keep diffusing. All the time it's moving through this tubing. Diffusion of wastes occur. looking at this, so for example, what if your sodium levels are 160 in the blood and the isotonic level is 140? What'll happen? What will happen to the sodium? Is it going to move from where? So that means it's going to move. Is it going to move out of the blood or into the blood? Out. So can you see that the salt would go, salt would begin to move out because it's 160 in the tubing, 140 in the fluid. So it's going to go from high to low. What about potassium? Potassium also. In this one, potassium would move from high concentration to low concentration. What about calcium? Here to go in. Do you see that the calcium's 125 in the dialysis fluid and only 1.2 in the blood? So calcium would actually go this direction. So some calcium would move in. Bicarb? In. See how the bicarb's 34? So it's going to move this way. So I would have bicarb would move this way. Magnesium. Out. Magnesium would go from 1.5 to 0.5. Chloride? In. Going across. So those arrows I'm doing all the way across just underneath that tubing. Doesn't mean it's going into the tubing, but it's just going that direction. What about glucose? So there is nutrients in the dialysis fluid. So if you look at this person, if their glucose is 4.5 in the blood, but 5.5 in the fluid, that means that glucose is going to end up moving in. So glucose will move into the blood. So this keeps happening. In fact, this dialysis fluid, as wastes begin to build, you would think that would slow diffusion, right? Because high to low till it's equal. So the way they make sure they try to get as much as possible is they're constantly replacing the dialysis fluid. So there is like fluid coming in, fluid draining off. So that keeps the dialysis fluid isotonic. So by constantly feeding in fresh fluid, draining off the used fluid, that's going to help to keep the dialysis fluid as an isotonic condition. And that is going to then, after the 4 hours, what should the concentrations be? They should be equal. So after 4 hours, this moving in and out, after 4 hours, the concentration of blood in the dialysis fluid should be the same. But now think about this. Your blood is filtered by your kidneys 35 times a day. Somebody that is on dialysis, their blood is filtered 3 times a week. So think about the change. You fine tune because your kidneys are always picking out water, salts, and waste that's like in excess. They don't have that option. So before they go to dialysis, how do you think they feel? Crappy, which is why they're super cranky, okay? Because their balance is all out. They have a lot of waste. They may have a lot of water. They might not have enough water. They might have a lot of salts. They might not have enough salt. So all of these are out of whack. And in 3 to 4 hours, it goes, okay? Everything out of whack gets like put into place. And how do you feel after?
They still feel like
they're
crap.
exhausted, okay? Typically after this, you're tired because you've like, it's like your tissues are suddenly like back into place, but it's just not, it's so abnormal that most of the time these people go home and sleep for 4 or 5 hours.
How did they not pass out during this?
Yeah, well, it's why they do it over 4 to 5 hours. They don't do it super, super fast. You don't want to do it, but so rapidly. It's just like the same thing with hydrating somebody, right? So you, well, and there is so much about diet and everything else that goes along with this, but this, one day you do dialysis, the next day, that's like when you go grocery shopping, that's when you go to your doctor's appointments, because then the next day, you go back to the dialysis center. And then the next day is the day that you feel okay, and then the next day you're back at the dialysis center. So this is something, and how long do some people do this? Their whole life. There are some people that have been doing dialysis for 25 years. Because of lack of kidney function. So then people are like, well, why don't they just give them a kidney? So 1, it's not that easy to do a kidney transplant. And 2, the odds of a kidney lasting more than 7 years is slim. 7 years is about the lifespan of a kidney transplant. So all of those factors kind of come into play, how old you are, what other health issues and everything else. But this is where osmosis and diffusion is like really used to try and maintain some quality of life for these people. Okay? All right, so that finishes 8. I'm going to give you, on your way out, 1, write all your answers on this. Please try to write legibly. Please. Please. Like, if I can't make out what it is. This is due the Tuesday that you come back. Everybody knows we have no, campus is closed tomorrow. We have no classes next week because it is our spring break. So the Tuesday you come back from class is when you need to turn this in.